Hi, so today I'm going over the urea cycle. This is a crucial cycle used in the degradation of amino acids. After the nitrogen group is removed from an amino acid by transamination, glutamate is formed. But alpha-ketoglutarate needs to be regenerated. This is accomplished in the glutamate dehydrogenase reaction, which releases the nitrogen as ammonia. However, ammonia is quite toxic and needs to be removed. The ammonia is scooped up immediately by the urea cycle in the liver, which converts it to a much less toxic metabolite called urea. Urea is released into the circulation, then eliminated through the kidneys. The overall reaction for the urea cycle is ammonia plus bicarbonate plus aspartate makes urea and fumarate. So now let's get into the steps of the urea cycle. The first step is to fix ammonia to carbamyl phosphate using bicarbonate and 2 moles of ATP. This is done by the enzyme carbamyl phosphate synthetase. Once carbamyl phosphate is formed, the carbamyl group is transferred to the amino group of ornithine. Ornithine is a non-standard amino acid similar to lysine. This is done by the enzyme ornithine transcarbamylase and forms citrulline. All the reactions thus far have occurred in the mitochondria. Now citrulline is transported through the mitochondria to the cytosol to complete the next few steps. The third step is to bring a molecule of aspartate and ligate it to citrulline to form arginosuccinate. And this is done by the enzyme arginosuccinate synthase and uses one molecule of ATP, but in this case AMP and pyrophosphate is released. Now since AMP is produced, this is equal to the use of 2 ATP equivalents, since AMP will have to be converted to ADP in the cell. So in total, the urea cycle uses 4 ATP equivalents. Another important thing to note is that the aspartate is needed to contribute the second nitrogen in urea. The first nitrogen is from free ammonia, but the second one comes from aspartate. Now, after the creation of arginosuccinate is cleaved to form arginine and fumarate, and this is done by the enzyme arginosuccinase. After arginine is formed, urea is released by hydrolyzing arginine using the enzyme arginase, and this regenerates ornithine, which goes back into the mitochondria and is used in subsequent steps in the urea cycle. Now I hope that you notice that there are some TCA cycle intermediates in the urea cycle. Thus there is a link between the urea cycle and the TCA cycle. The fumarate that was produced in the 4th step can be converted to malate by fumarase, and malate can then be converted to oxaloacetate by malate dehydrogenase, and oxaloacetate can be transaminated to form aspartate that was used in the 3rd step of the urea cycle, and this regenerates fumarate. It's a very nice fitting cycle. I hope you have gained a basic understanding of how the urea cycle works from this video. Thanks for listening.